Today, the topic of discussion is polysaccharides. Polysaccharides, the sugars, those sugars which yield more than 10 monosaccharides on hydrolysis are called polysaccharides. There are two, there are two types of polysaccharides, homopolysaccharides and heteropolysaccharides. Homopolysaccharides and heteropolysaccharides. Homopolysaccharides, it is a polymer of single monosaccharides units, while heteropolysaccharides it contain other groups other than carbohydrates. Homopolysaccharides also called as homoglycans, while heteropolysaccharides also called as heteroglycans. Examples of homopolysaccharides starch and glycogen, while example of heteropolysaccharide is mucopolysaccharides. Now I will discuss homopolysaccharides. It is a polymer of single monosaccharide units. First of all, starch. Starch is a polymer of glucose. It is used as storage food in many plants. It is present in leaves, stems, and roots. In seeds, it is present in greater concentration. Starch granules are made of concentric layers of materials. They are consist of two polymeric units, amylose and amylopectins. Solubility of the starch. It is insoluble in cold water, while when heated, water taken and swelled and make paste or gels. The reaction of starch with iodine. With iodine, it gives blue color. Amylose gives deep blue color, while amylopectin gives blue-violet color. The difference between amylose and amylopectin. Amylose is 15 to 20% present, while amylopectin present in star 80 to 85%. Amylose has low molecular weight, while amylopectin has high molecular weight. Amylose is soluble in water, while amylopectin is insoluble in water. When heated, it absorbs and swells up. Amylose is unbranched and straight chain, while amylopectin is branch structure. The linkage present in amylose is alpha 1,4, while in amylopectin main stem, main branch, main straight chain contains alpha 1,4 linkage, while branch contain alpha 1,6 linkage. Starch, when in the presence of amylase hydrolyzed into maltose with small quantity of dextrin, which then converted into glucose with the help of maltase enzyme. When starch is hydrolyzed in the presence of acid, it is directly converted into glucose. Glycogen. Glycogen is a polymer of D-glucose. It is reserve carbohydrate of animal, therefore it is called as animal starch. It is present in plants which have no chlorophyll like fungi and yeast. In higher animals, it is deposited in liver and muscle as storage material. Glycogenesis. Glycogen formation basically from the glucose. Glycogenolysis breakdown of glycogen to form glucose. It gives deep red color with iodine solution. Then inulin. It is a polymer of D-fructose. It has low molecular weight. It is present in bulbs and garlic. It is absent in human. Therefore, it has no importance uh, in human. It is has its biomedical importance. It determines the rate of glomerular filtration rate. It is used for the estimation of water body volume. Then agar. It is a polymer of galactose which is sulfated. It is used as laxative in constipation. It is the biomedical importance of agar. It dissolves in hot water as cooling it forms gel. It is used as agar plate for culture of bacteria. Then cellulose. Cellulose is a polymer of glucose. It is not hydrolyzed with dilute acids, while in the presence of concentrated acid, it yields products. It is the main constituent of supporting tissue of plants. In animals, it is digested because of the gut bacteria are present. In human, it is not digested as gut bacteria are absent in humans, which break down the cellulose. It is when it is added to the bulk amount in in bulk amount added to the intestine. It is used for the elimination of indigestible food. Then dextrans. Dextran, it is a polymer of D-glucose. It is synthesized by the action of Cornstalk mesenteroids, a non-pathogenic gram-positive cocci bacteria. Its biomedical importance is it is used as plasma expander in case of hemorrhage. In it, it increases the blood volume and stay for a long time in the blood. Its disadvantage is it interferes with the cross matching and grouping of the patient. Therefore, it is recommended that before hemorrhage, before the it is administered into the patient's blood, the blood samples should be collected before the administration of the dextran. Now, dextrin. Dextrin is basically when starch is partially hydrolyzed into maltose in the presence of enzyme amylase. Dextrin are formed in small amount.